Welcome, my trailblazers. Welcome, digital pathology trailblazers.
Learn about the newest digital pathology trends in science and industry, meet the most interesting people in the niche, and gain insights relevant to your own projects. Here is where pathology meets computer science. You are listening to the Digital Pathology Podcast with your host, Dr. Alexandra Zhurov.
So, thank you so much for joining. I'm Dr. Alex. I'm a board-certified veterinary pathologist, and I run the Digital Pathology Place. It's an online platform about digital pathology and medical AI. And today is our weekly DigiPath Digest journal club, where we discuss the newest papers about digital pathology and medical AI, mostly digital pathology. So I discovered something cool that I can actually have a different layout like this. These little things give me joy. But let's start with our first papers. We have 4 today. And so we have computational pathology as a mechanistic discipline from morphology to molecular data. And we also have the infographics. The infographics were hit last time because the abstracts, as we said last time, they don't give justice to the publication. And then I listened to them on podcast from Notebook LM, and there's so much more to the paper than the abstract. But in this particular one, computational pathology as a mechanistic discipline from morphology to molecular data, the authors say that pathology is undergoing a shift from morphomolecular interpretation toward computational integration of molecular mechanisms encoded in tissue architecture. This is really high-level explanation, but we're going to dive into it. So, such as morphology-driven molecular inference may enable biomarker prediction and potentially generate therapeutic insights directly from routine histology. So, what are we talking about here? Here, we're talking about molecular predictions from H&E. So, but there is a caveat that, hey, why do we even believe these predictions? It's just like a correlation. So they explore that actually. The paradigm has important clinical implication for quantitative biomarker testing, patient stratification, design of digital biomarker-based clinical trials. And they are mentioning a digital biomarker. We're going to mention it as well. So the most current artificial intelligence models remain correlative. We just mentioned that. And the clinical impact depends on rigorous validation, integration into workflows, and ethical governance. We have such a good paper at the end about AI literacy. So definitely stay till the end. And if you can't, watch the recording. Fellow veterinary pathologist of mine wrote this one. But when I saw this ethical governance, that's why I remembered this paper, because there is a part on that as well. And obviously addressing these challenges will be essential for computational pathology to turn to mature, clinically meaningful discipline and not just a bunch of like single applications. Let's have a look. But the first one, what I like very much here is they have these different levels, right? And we are starting with level 1. Level 1 is our morphology layer, what we see, right? Then we have level 2 is the AI computational layer. So this is like the molecular prediction from H&E is happening with this computational layer, but what they say, we also need the ground truth. And I am not talking about annotations by pathologists. We are talking about spatial omics grounding the AI. So we have spatial transcriptomics, proteomics that you put on top of the on the tissue, right? You can see what transcripts, what proteins are there. And then when you query the AI, our level two, where did you find this information? And let's say the information was found here. It's a stroma, let's say in the stroma, and we have a ground truth from spatial omics in the same region, stroma, then we have an indication that it actually took the information necessary for the decision from the right region. Is it like 100%? No, but that's as close to 100% as we can get. So now what we can do, we can get from these subjective categories also, that's kind of a separate thing, because now we used to score IHC with these buckets, 0, 1 plus, 2 plus, a bunch of markers, IHC markers is still being done like that. But actually it's a continuum. And you have to like arbitrarily make the cutoff where 1 plus ends and 2 plus starts. Maybe it's 1.5, but you don't have the visual capacity, even capability, physiological capability to do that. But the computers have that, and we can have quantitative continua. They are referencing that now, I hope, famous, and if not yet famous, there's going to be a webinar about this, quantitative continuous scoring for the TROP2 marker that stratified patients, and this scoring is calculating a ratio from the membranous staining of a cell to its cytoplasmic staining, and it also normalizes the membrane staining, and there's going to be a webinar about this with Roche team. And in the meantime, let us go to the next paper. Let's do this one. The next paper is Advances in Digital Cytopathology and Artificial Intelligence Applications. This cytopathology is something that even though I'm an anatomic pathologist, so I look at FFPE, for formalin fixed, paraffin embedded, and not really cytopathology, I'm thinking the preparation of cytology specimen is so much easier, but the digitization is so much more difficult. But also, the first applications of digital pathology were in cytopathology. So what happened in between? And this is what this paper is addressing. So let's look. It's a review, and the authors are Saturwar, Parwani, and Lee from Ohio State. Let's look at the abstract. Cytopathology is the first field of pathology in which AI models were successfully developed, like FDA cleared models and there was they were commercialized for routine clinical screening of cervical cytology. And this has been in place for the past 2 to 3 decades, I guess, depending on the applications. I was wondering, is it 2 decades or 3 decades? But probably depending on the application. And the AI applications for non-gynecologic cytology. So this was cervical cytology. This is Pap screening, Pap smears, cervical cancer screening. But non-gynecologic cytology, this has just begun. The problem is the variety of cytology specimen types and preparations with associated unique characteristics presents technical challenge for the complete digitization of the cytology workflow. And this is a very diplomatic way of saying they look so bad sometimes compared to an FFE specimen. Like, they don't look great because you have cells, smears, and the level of interpretative effort when doing cytology is so much higher than when doing anatomic pathology. So that is the problem, because they are not thin enough, not even enough. The cervical cytology, they standardize the way they are creating these slides, right? They have the thin prep, which is centrifuging everything away other than the cells, the cervical cells that we're interested in. And then it's being like put on the slide in a little circle. So it looks really, really nice compared to the other specimen that can be like all over the place. But despite these challenges, few institutions have a complete digital cytology workflow, and technical investment have replaced conventional rapid on-site evaluation by different telecytology systems. So we're still doing live microscopy, but now it's telecytology. Some can be scanned, but most are being done with remote controlled microscope. And there is one system, Hologic Genius, and that is cleared, not approved. I checked. But this is like, FDA terminology, regulatory terminology. This is the only one that is cleared by the FDA for routine clinical screening of cervical cytology in the United States. So we're still in cervical cytology, right? The beautiful looking thin prep. And here, the recommendations for AA validation and best practice guidelines for digital cytopathology are currently being developed. There is a task force in Digital Pathology Association, and there are technical advancements in automation for sample preparation, ROSE, this rapid on-site evaluation, using telecytology. There's automation for screening of gynecologic and non-gynecologic cytology specimens, automated quantification of biomarkers, quality control, and this is what this review is talking about in detail. Now let's move to our infographic. So what is happening here? We also have level 1, which was the established foundation, the gynecologic cytology. Then we have level 2, what is currently happening, digital infrastructure, telecytology for ROSE. I don't know why they did this MRI picture for telecytology. Maybe because they're doing something with the patient and the patient needs MRI and then they do the evaluation. So maybe that's why. So in this level 2, we have evolution of virtual telecytology, digital diagnostic solutions, establishing validation standards. This is where we are right now. This is what we can do safely. And then we can go to level 3 and expand our horizon to non-gynecologic samples and advanced AI. So the transitioning to non-gynecologic, that's going to be, it has begun, but it has challenges regarding diverse specimen preparation and unique characteristics. So check out how these cytology specimens look, and they are challenging for human eye. So if they are challenging for human eye, then they're going to be challenging for computer vision. But what is happening right now in this level 3, there is automated quantification and quality control. So specifically for cervical screening, there is already a workflow that has a triage built in, and there is a cytotechnologist looking at it, and then the pathologist for the ones that are suspicious. And now you can put AI on top of that and do 100% quality control of the negative samples and catch anything that actually was not negative, was false negative, right? And if we could achieve total workflow automation, that would be amazing as well. Let us move to the next paper. What are we talking about? We are navigating the labyrinth of hepatocellular carcinoma, leveraging AI and machine learning for precision oncology. And hepatocellular carcinoma is a significant global health challenge. We're going to see it on the infographic, how much they are predicting it's going to rise in the next 20 years. I was very surprised by how much. And then the problem here, therapeutic efficacy in advanced stages is often limited by underlying liver dysfunction and adaptive resistance. So this is a reviewing the landscape of molecular targets and combinatorial strategies, and they critically examine it. They focus on the transition from preclinical discovery to clinical application. They take into consideration the molecular heterogeneity, and what they say here is that computational paradigms are redefining target discovery and therapeutic stratification in hepatocellular carcinoma. And they see the role of AI and machine learning as integrative tool for translating high-dimensional multi-omics data into insights for HCC management. And also they describe AI-driven frameworks to analyze complex data sets derived from genomics, transcriptomics, proteomics, metabolomics, and epigenomics. So we are doing multimodal here very much. This is now relevant for cancer therapies, different cancer therapies, in this case, hepatocellular carcinoma. And they are also discussing can AI enable identification of novel predictive biomarkers, patient subgroups, and rational drug combinations. They acknowledge the convergence of AI with spatial transcriptomics, digital pathology, single-cell technologies. So pretty comprehensive review. It's going to be very clinically focused and oncology focused, but we're going to focus in the infographic on the AI part. And this infrastructure that we have, the AI infrastructure, it can be used for decoding tumor microenvironment interactions and spatial heterogeneity. And tumor microenvironment interactions, this knowledge about them is non-negotiable for modern immune oncology. And what they claim is AI-enabled multi-omics-driven approaches are instrumental in advancing HCC treatment. So let's dive into the infographic. Here, what I was surprised that they have this projection of 55% increase by 2040. Like, that's a lot. Why? So they say that they are diverse etiologic drivers, viral, alcohol, metabolic, and non-alcoholic hepatic steatosis, and also aflatoxins, right? So different things, different molecular drivers of these problems of the cancer itself. And they have this timeline about management of this disease, where we are now present 2020. What we have is immune oncology era combination therapies. And then in the future, they are envisioning gene therapy era with different gene therapy modalities. I want to focus on is this lower part. Let's do this here. There is this AI-driven precision pipeline. What do they mean by that? There is the data acquisition part. I'm wondering if this is even anatomically correct. I have to, well, there is a gallbladder. That's a digression about the quality of the infographic. I'm going to show you where AI did some funny things. So we have data acquisition, right? Clinical data, multi-omics, radiomics, and then we have this AI architectures and training. We have convolutional neural networks that are good for imaging. Then for multimodal, we have some other deep neural networks. And we can, then we have unified molecular profile, and we can do a lesion detection with this, and also predictions, so-called virtual biopsies. And the decisions that we are trying to get from this is, as we said, lesion detection for maybe radiology or pathology as well. What's going to be the post-transplant recurrence, immunotherapy response, and 5-year survival prediction. So this is what would be amazing to achieve with this AI-driven precision pipeline. Let's look at our last paper. Last paper, I left the very actionable thing as our last thing to discuss, because that is basically gonna affect all of us, me including, all the time. What we have here is the curriculum framework for artificial intelligence literacy in veterinary education. And veterinary education, you can basically substitute this with anything else, with medical education, with, well, there are specific things that are unique for veterinarians here, and the authors mention it. So the authors, Yiting Huang and Candice Chu, and I want to show you, I want to highlight that last author. Who is Candice Chu? Candice Chu is a fellow veterinarian, fellow veterinary pathologist. She's a clinical pathologist, and she's an assistant professor at Texas A&M University. We briefly met. We were like colleagues at some point, and then so she entered at the place where I'm working, but then she did so much amazing work in veterinary medicine on AI literacy, AI applications. And I also had her on the podcast. Let's see if she's going to join me again about this particular paper. But she is so involved in teaching AI to veterinary professionals. So I just wanted to highlight her because I think her work is really remarkable. And also, not only this, she's very active in promoting her work through social media platforms. On Instagram, you can find her on Instagram. You can find her on LinkedIn and other platforms. Let's see. She's LinkedIn, Facebook, Instagram, obviously Google Scholar. You can have a look at what she's publishing. But let's go into the paper. And then we have an infographic about the paper as well. What the authors are saying in this particular paper is that AI is rapidly evolving very fast, and it is increasingly incorporated into veterinary medicine. So veterinary medicine, let me tell you how it works. It's a lot less regulated than human medicine. So application of different things are gonna happen faster there. So digital pathology, the first system deployed in like a corporate environment, was at the veterinary pathology laboratory at scale. They tested everything, and then only 2 years later, they actually set up all the trials and filed for FDA clearance of the system or approval or whatever that was. So the work, the cutting edge technology application in veterinary medicine is happening a lot faster because there's less regulations, but the literacy of veterinary professionals in this space need to be really high because they are still liable for whatever the outputs of AI are. And they are highlighting this very much in the paper and in their curriculum. So given the demand for integration and the accountability placed on licensed veterinarians, there is an urgent need to integrate AI literacy into the Doctors of Veterinary Medicine curriculum. So they're talking about US, but it is applicable everywhere. 5 core modules for a comprehensive AI literacy curriculum for DVM students that can serve as a foundation for institution-specific course development. So, you can take her framework and basically develop a course at your university. And that's what they would like you to do. They urge institutions to provide the resources outlined in this article to facilitate the development of AI literacy courses in veterinary schools. And let me tell you, it's not like a superficial thing. It's deep. I believe that if I would take part in this course, I would learn a lot, even though I'm here discussing papers with you. So let's move to the infographic because it's super cool how they design it. And really, it is deep and it prepares you. So here we have it, 5-module framework for veterinary education. This is from April last month, right? And I saw it on LinkedIn on Candice's profile, and I'm like, I need to discuss this one. High quality work, great design curriculum. So again, we have layer 1. Layer 1 is, they call it the physiology or fundamentals of AI. What are we learning here? We're learning about the architecture of different frameworks, right? They are discussing how convolutional neural networks see pathology, and because the clinicians must understand, like they compare it to cellular physiology, they must understand the normal, what's happening in the cells to see the abnormal. And basically this is the premise. Like if you're going to be using these tools and if you're going to be liable for what the output is, you need to be able to detect when it's not working well. How are you going to detect it? You need to know how it works. So this is module 1. Then layer 2, module 2, the mechanics, machine learning, and evaluation. And in this course, they actually at Texas A&M, they make students like design an image analysis application. So what they are doing here is hands-on with YOLO. YOLO is an image analysis framework. You only look once architecture for real-time image recognition and classification. They use the canine lymphoma application, canine lymphoma as the entity that they're working with. I promised you that I'm going to show you where the infographic didn't go so well. Here, like, what is this? FGTL? I assume it was supposed to be fail. Let me show it to you. This is just the fun part. What did Notebook LM do not so fantastic on this infographic? Here, this one. And there is something else here. I don't know what this image is, but also the text says research bites, and here's like research OHES. So, trust but verify. And that's very fitting to this particular AI literacy paper. Okay, that was just me digressing on my Notebook LM output. Let's go back to the content. So we were layer 2, right? Then what they say, because you're going to have a company coming to you and saying, oh, we can do this, that. So you actually have to be able to integrate the shiny brochure. And then you have to check the performance metric versus real world bias. And it's very evident in veterinary pathology. You can see like this dog is thin and doesn't have so much fur. And the other dog that the image or whatever the algorithm was deployed on samples from another dog or images from another dog looked totally different. So like you have these breeds of dogs or different animals. And if it was developed on one, there is no guarantee that it's going to be working on the other one. It's similar to different demographics in human medicine. And then what are we doing in layer 3, module 3? And there is the large language models and prompting. There's interacting with AI. There's going to be a lot of text AI because there's going to be a lot of AI that is going to be workflow focused, speaking focused, like ambient scribe is one of them, or there are veterinary versions of ambient scribe where you talk to the owner of the animal. The clinical notes are being created from this conversation. So they emphasize that large language models are probabilistic autocomplete. They teach you what it is. It's not a person that you're talking to, right? And also they dive into so-called vibe coding and agents. I saw that and I'm like, they are cutting edge because they're doing what's like really happening right now. These CNNs were happening like 3, 4, 5 years ago, maybe. That was the beginning, right? And then now we're talking about vibe coding. So vibe coding is just like speech coding. You can create apps talking to an app creation platform. You don't need to know how to code. You probably need to speak English, but I bet there are already vibe coding platforms in different languages. So you can like code an app for your practice, but then you're still somebody liable for the outcome of this app. So they're learning that, and AI agents is the workflow orchestration or orchestration of different steps of AI. So now we've learned all these things, even the newest hypes, vibe coding, we can make an app. But what about the guardrails, ethics and liability? This is covered in module 4. And here I want to highlight something. There are no FDA pre-market requirements for this AI software for veterinary medicine. For veterinary drugs, veterinary medications, there's normal like FDA approval, and this has to go through a specific drug development process. In human medicine, there's this framework of software as a medical device. It's a medical device, and then FDA regulates these medical devices when they become products. In veterinary medicine, nope. That means for the companies that are providing this, fantastic. They can that's their playground. For the veterinarian, very, very high liability of something that maybe is not that thoroughly tested yet, right? So they emphasize the human in the loop requirement because the Veterinary Medical Association in American Veterinary Medical Association, so they are talking about the US, says AI is strictly assistive. The licensed veterinarian is 100% legally accountable for every diagnosis and medical decision, right? So if something goes wrong, your beautiful vibe coded app, if it didn't work well, it's your fault. So you need to learn about ethics and liability. And then layer 5, module 5 is the research and academic integrity for scientific writing, for synthesizing. And what they say about this layer 5, synthesis, research academic integrity, and the context window danger. We probably already heard about hallucinations multiple times, fake citations. There were papers pulled from Nature or high impact, I don't know if it's Nature, but very high impact factor journals for fake citations. But also another thing is blend conflicting research papers into a single false conclusion in the document length, if the document length exceeds their context window. So context window is how much information can a large language model like see at the time, how much can the transformer kind of like oversee at the time, and if the length is longer, then it only sees parts of it and is drawing conclusion on the part. And they are also teaching different tools, AI-enabled literature synthesis. What they say there is that staying on top of literature manually now is very challenging, if not impossible. And they do use tools like Notebook LM, which I am using as well, research bytes to efficiently manage the rapidly expanding volume of veterinary literature while maintaining authorship integrity. Candice also has this writing framework, AI-powered writing framework she gives workshops on. And if you go on her social media, you're going to find her framework. You can download it. So I love this one. I'm wondering if anything like that is happening in medical colleges or medical associations. Yes, probably yes. If you know like a specific course, you can send me a link in the chat here, even if you're watching the recording, because I would like to just learn more. Okay, what is the program, official program by official associations? And I'm going to do my research as well. But if you know something, if you have taken some courses that were good, I obviously have my Digital Pathology one-on-one book that is for those who are starting the digital pathology journey. And there is a chapter on AI in the new version that is almost finished. By the way, if you want the new version, get the book. The book, you can find it on digitalpathologyplace.com, and you go on digitalpathologyplace.com and you're gonna there's gonna be like a pop-up. You're gonna find this book. You can download the PDF for free, and then I'm gonna let you know every time I go live. But also you can see this edition is from 2023, and I'm currently working on 2026 edition. It's almost ready. The PDF is going to be the first one to go out. So if you're already on the list, if you have the book, then you're going to automatically get the new version. I will be bragging about it on social media as much as I can once it's out, but you can already be on the list to get the new version. Thank you so much for joining me today. I appreciate you so much, and I talk to you in the next episode.